everybody. I am Karina Sargsyan, and I am very happy to moderate this panel on the environment and health and how human health is getting actually affected by the environmental problems. Please welcome with me. Oh my God, I don't see my panel at the moment, which is very strange.
We can see you. We're both here.
Really?
Yes.
Yeah, perfectly well. Hi.
Oh, hi, Lucas.
Hi, Karina.
Hi, Sis. So I'm very happy to, till I find my panel back on my many windows open, as actually women do, not structured. I'm very happy to welcome my fellow panelists, both my friends, and I'm very happy and honored to call them friends. Dr. Zidis Koslakidis, the head of the IARC Biobank, and Professor Lucas Kenna, who is actually one of the most important people on pathology and microplastics in cancer in Europe. So I'm surrounded with very intelligent people, and I'm a little bit of intimidated, but nevertheless, we're gonna speak about how environment is affecting our health. And I want to start with Zizis and ask him, what do you think? How does air pollution, especially gases eliminated that are harmful, increase the global prevalence of any kind of diseases, including respiratory, cardiovascular, but specifically cancer?
The short answer is yes, with a lot of asterisks. So let me explain. Just as an introduction, hello to your participants. This is Koslakidis. I'm a microbiologist by background, so you understand the lens that I will be wearing in this discussion. And at the International Agency for Research on Cancer, part of the World Health Organization, as the head of laboratory services and biobanks. So, having said all that, so the pollution is something that it's hugely concerning, because we have different levels of pollution, different types of pollution, and different exposures. And with all these differences, there seems to be a health effect at the end of the road. Now, the question that we often ask is something more harmful than something else? So I think to be realistic, air pollution we try to avoid or mitigate as much as possible, but to be always able to avoid it, it's not really that practical, especially in the highly urbanized environments that we live. So what we do is we try to identify the types of pollution that are the most harmful, that where we can have the very specific cause and effect relationships with particular disease conditions, and then try to produce guidelines or initiatives to reduce those. So to give you an example, there is very good correlation between levels of air pollution in cities and levels of asthma in children. It's not cancer, but it's a chronic condition that could be avoided for a large portion of the population that develops it. And it's a lifelong condition that needs to be managed. So it doesn't kill you, but it exerts an influence over the rest of your life. And that's significant enough because it reduces or affects your quality of life to some extent. There are certain types of exposures, in particular toxic exposures or exposures to toxic substances. For example, some of the industrial diluents would classify in that category, where they would have toxic effects. And if not properly protected, they may relate to cancer. So it's a complex relationship. We like to say this is black and white because it's helpful to take a decision. It's not so black and white, but there are many aspects to consider. The more we search, the more we find. And I think we'll find at the end that most of these types of exposures at the end will have some impact on the overall health, whether it's direct, indirect, cumulative, and so on. Thank you.
Well, thank you very much for giving me a platform to ask so many questions. So the first thing I want to say that we imagine, for instance, the air pollution or microplastics, just black and white, as you said, but one of the sources of microplastic is inbreeding microplastic that usually the common population doesn't think about that because we think about the seafood, we think about the bottled water and so on, but nobody thinks that we
No,
inhale
even
plastics.
toothpaste, most of
Exactly.
toothpaste had microplastics until recently.
Exactly. So my next question goes to Lucas. So what role do microplastics play in disrupting all the health system, including the endocrine system, including cancerogenesis, including long-term health implications? I know it's a big question, but we would like to hear from an expert. What is 1, 2, 3, 5?
First of all, thank you very much for this kind invitation to talk to you in this very prominent venue. And my name is Lucas Kenner, as said, and I'm a pathologist. I'm here in my office at the Medical University in Vienna, but I'm also teaching at the Veterinary Medical School in Vienna and guest professor in Umeå in Sweden. And one of my topics since more than 3 years now is microplastics in health. The reason why I focused on that was basically a colleague of mine, Philipp Schwabel, who published just 4 years ago here that microplastic was found in the human stool. And I was really surprised when I heard that, and he told me that, and I was then going into the PubMed and was really shocked that I couldn't find even one publication on microplastic and health. And then I basically initiated a consortium called Micro1, which is now basically we are almost 3 years out and being funded without even having a single piece of data except the ones of Philipp Schwabel, who is now in London actually. So to answer your question, I mean, it's difficult for me now because there are so many possibilities that microplastics can enter the body, because as Tizi said, we inhale it by pressing, we swallow it by taking our food and drinks, and it also can enter via the skin or via the skin and mouth and so on, so via contact, and also into the brain via I've just learned recently by a publication which came out just a few weeks ago, via the olfactory nerve, for instance. So we are exposed by a lot of different ways, and not only we, so the whole ecosystem, because
Lucas,
the
I'm
plastics
sorry, you said olfactory nerves?
Yeah, through the nose, and it can enter via the epithelial layer of the nose, and then it enters into the olfactory nerve. And I was surprised that it's possible, but it was just published by a study of a Brazilian group, which showed that in patients, when they investigated, that obviously
we knew that we can inhale and it goes to lungs, but we didn't knew that it affecting also the nervous system. Oh my God.
Yeah. We published 2 years ago, no, almost 2 years ago, that the plastic particles can enter the nervous system via, blood-brain barrier, and we were surprised because we just fed the mice with plastic particles for 2 hours. So after gavage, we already saw particles all over the body, including the brain, and then we teamed up with a colleague who modeled the blood-brain barrier, and we found out that the plastic particles cover themselves with cholesterol molecules, and thereby, we call it Trojan horse phenomenon, and thereby these particles can enter through the blood-brain barrier into the brain, and this can happen also in other body barriers. So obviously, these particles, they have different, possibilities to cover themselves and to mimic, other by covering themselves with, let's say, cholesterol or maybe albumin or whatever molecules are there which can bind to the surface, because the plastic particles have different properties, one of which is that they have different charges. They can be positively, negatively, or uncharged, and thereby also they can attract, other molecules like amino acids, nucleic acids, which are differently charged, and then these are possibilities. But there are also other very interesting properties like their morphology. This can be very different, and so we're working with polystyrol particles, for instance, which are perfectly roundish and smooth surface, but there are other particles which look more like rocks or snowflakes, and they have very different properties which are surprising and can change the properties of the particles when they enter to the body. And we are really at the very initial stage of this kind of research, because obviously not a lot of people have went into this field, and we are here basically some kind of pioneering this investigation of what is the role of plastic particles, and I cannot answer it really to your satisfaction, because especially when it comes to human tissue, there are just some studies out describing how much particles are found in different tissues, in human tissues, but this doesn't tell you what's the role of the plastic particles regarding long-term effects. I mean, there was one study just recently, because just 2 or 3 months ago, in prostate cancer, that particles can be found in prostate cancer more than in the surrounding non-malignant tissue, like about 100 microgram per gram tissue. So that's just an average, which I say.
That might relate to cholesterol, because the cancer tissue
Sure.
is well known. Yeah. But this is absolutely fascinating. Can I ask you a question, Luke?
Yeah, absolutely. I guess we are here to talk.
I knew that's gonna happen. I knew that's gonna happen. Yes, please.
It's just because the microplastics, I mean, we produce them, but then there are degradation products as well. So there is 100 different ways in which we can result to them. So is there a way to predict from an original substance what microplastics may be derived, or it's anybody's guess?
You mean what is the source of the plastics, or
right, exactly, yes.
Well, that was the biggest issue we had, at the beginning when we started, that I'm pathologist, and I'm used to analyze tissues and also use different microscopes, including electron microscopy. And when we wanted to detect these particles, the only thing which I really could use, we have here everything, including sequencing and proteomics, was the electron microscope, because with the electron microscope you can visualize plastic particles, but with other technologies we need the experts like Raman spectroscopy or infrared spectroscopy, different technologies which we don't have. So we were just recognizing that we were a bit helpless in the medical environment, and we teamed up with chemists and chemical analytics technology, also computer modeling to predict what happens when these particles enter a cell or enter the body and come into touch with body barriers. And we are just trying to interpret the findings we have based on this kind of technologies. So therefore, I'm very happy that you asked me this question. So of course, that could definitely be possible, yeah, and I would say that's or maybe even likely, yeah. But I cannot, answer your question to say it is like this, because we are doing experiments with polystyrol particles, also with PET particles. So there are different subgroups of plastics, but we are influenced by really a lot of different plastic sources, which we can't cover at one time, because experiments become not reproducible if you use, I don't know how many particles at the same time. So we're doing one step after another, and this is an incremental progress, of course, but at the end of the day, we are influenced by, I don't know how many different sizes, shapes, and different properties and compositions of plastics. And I didn't even talk about the additives. We have about 14,000 different additives which give the plastic their shape and their behavior and so on, when you talk about the original products. And only 1% of the microplastics which we basically which we take up are intentionally added by the industry. 99% are basically degradation products, or a result of degradation, or UV light or whatever, because these particles are not produced by the companies, but have been originally your toothbrush or your bag, your plastic bag, or some kind of plastic dish you basically put somewhere into the environment. And these products, they decay and decay and decay. And then at the end of the day, if they are smaller than 5 millimeters, we call them microplastics. And if they are smaller than 1/1000th of a millimeter, let's say 1 micrometer, then we call them nanoplastics. And this, the smaller these particles become, the more biologically relevant we say, they are. But even this is not totally clear, because some people say in the gut, the microbiome is influenced more by the microplastics and not by the nanoplastics so much, because the bigger surface of microplastics may be more important. So these are just a little bit of introduction into the complexity of the field.
But it means we need to change our view, because when we talk about plastic, it's generally something neutral, no? But actually it's something that's biologically interactive, and
yeah, exactly.
Not that useful in terms of the interactions.
Guys, I love the discussion. I really love the discussion, but keeping in mind, not doctors are watching us, but the general population.
Correct.
So we need to maybe a little bit scare people to understand how important it is to keep in mind of less usage of plastic and so on in one hand. In other hand, we have, as we discussed, a huge gap in knowledge. So, and this knowledge gap is not only in the material sciences, like in the plastic and so on, but also in the side of the medicine and medical research, because how to find that plastic? How to know that plastic caused something? So keeping in mind that we are speaking for non-researchers, non-medical people, this is, what are the main knowledge gaps you think that we have in environment-based, pollution-based, and plastic-based changes of health? I know it's a difficult question.
Yeah, it's more where to start.
Exactly.
So the Yeah, I promise I won't scare people with acronyms and that sort of. The whole idea is so we have knowledge gaps and we have white spaces. I try to separate things in this way
Oh,
to
what is
try
white
and
space?
so a knowledge gap is where we have some knowledge but not all of the knowledge. So we know we have a
Oh,
gap.
okay.
So for
Gotcha.
example, we've characterized a lot of toxic substances, but not all of the toxic substances in the industry. So there is a gap in our knowledge. A white space is we don't really know how the biodiversity affects human health because it takes it's got too many of these knowledge gaps to put together as a puzzle piece. The best way I can describe. So I think in terms of the plastics and cancer anyway, that's where I'm looking at it from. We're still in the very, very early stages. So we do a lot of research to try and understand what are the effects, but this research takes a long time. So people are exposed to a particular substance in their environment. So it may be plastic in the water they drink on, maybe plastic in the food they eat. And you have to follow these people, the same people, with regular interaction over the years and see who of these people will develop a disease, cancer, for example. Will they develop it earlier or later than the general population? Will it be more aggressive than in the general population? Would it be more predictable? And these are the typical sort of questions we would ask. But to do so, it needs 20 years, 30 years. It needs for things to start from when you're at the stage of being healthy and being exposed to all the way to the stage you have developed the disease and we have studied the disease as it's developed within an individual.
But how you norm the exposure? I mean, plastic is everywhere. We even inhale.
Well, that's exactly true, yes. And it's incredibly difficult thing to do. So there are proxy, let's say, so there are you create mini environments where you can um, estimate the exposure. You can have model organisms, so you can do it in mice, which accelerates the speed of time, but it's not always transferable what we find in mice to humans. And you can always do it with a lot of environmental monitoring. So people can have wearables like paper patches that are sticky and things will stick to them, and then you measure it every now and then you have some measure of the exposure of the individual. So it's a very long-winded process, and it takes time. And I can understand that often individuals will feel a frustration that this problem has persisted for many years and nothing is being done. It doesn't mean that people are not looking into it. It doesn't mean that research is not being done. It's just the process is very slow.
Understandable. I mean, by researching what is happening, I found so many different. That's why I asked data. So from 10,000 to 52,000 microplastics annually. So 10 to 52,000 is 5-time difference. Well, 10,000 is already a huge number. If we are annually exposed 10,000 times to microplastics, is already a huge number. But if you look through different sources, you have 5 times of difference. That's why I asked how you're going to measure how much the exposure was, because sometimes we know in medicine that the reaction of the body to an exposure is depending on the dosage. That's how we dose the medicine. So you need to know about the dosage too to understand the research.
Well, yes, in principle, but also you have synergistic effects. So you can have exposure that in isolation is not necessarily above a threshold to create a problem or initiate the development of pathology, but you can have a number of these exposures happening at the same time, and together they move biologically beyond the threshold where things will start happening.
Well, if we speak about so many different layers, there is a layer that is, I think, very important to at least kick start a change, which is the policy. So, Lucas, what do you think? What policy actions or community-based initiatives maybe are most likely to really reduce that exposure? Or do you think it is possible at all at this moment?
I think it's very difficult. We played this through, and we initiate also a clinical study at the University in Graz and in Lithuania, where we basically try to measure, the effect of fasting, plastic fasting, because that's what we are talking about, to reduce the plastic consumption by telling the people to, don't use only for 8 days, basically. Don't use toothbrushes. Don't buy plastic enwrapped in food enwrapped in plastic. Get rid of your plastic in the kitchen. So don't use plastic surfaces or plastic cups or whatever, and don't drink out of plastic bottles. Things like that, And then we measure basically the plastic content in the blood and in the stool and in the urine at the beginning and at the end of these 8 days, and try to find out, can you reduce the plastic contamination by basically changing your behavior and changing your habits. And we don't know the outcome yet. We just finalized the study, and we are evaluating, because we need to do we do the microbiome analysis by genome sequencing, so RNA sequencing basically, and so this takes some time. But we can educate the people, and what we saw is basically that people are very concerned and very much aware, and they keep also being aware after this period of time, when we basically educated them to do this. But of course, to know if plastic fasting really would lead to a change, in the contamination of body fluids or body contents, basically, and in the end also maybe lead to a reduction of the contamination of cellular structures or organs in the body. That's a totally different question, and of course, I mean, if you just see basically or measure the content of plastics in products like just bottled water, like last year, I think this was Columbia University just published a study on 4 different bottled water products, and how much particles are in one bottle of 1 liter of water. It's 10,000 of particles or even more. So that means that just in this bottled water, in just one of these products, you have more particles than we originally thought was the yearly doses of plastic. So we will see a lot of other data, maybe from highly processed food or so, where we know that the contamination is likely not what we assume, but maybe higher. Yeah, but we don't know what it means, and that's the big issue. So we can and of course you have to be careful, because the technical limits to measure plastic particles is limited into larger particles like 1 micrometer, but smaller particles really difficult to measure with the current technology. So you have to be careful in the numbers which are currently out there, and that's just some that you get some ideas that this is not an easy, problem of if you measure, contamination by a substance, it's much easier because then you can, this we know how to measure. But this particle problem is something in a different dimension, and therefore I'm very cautious to give you some numbers, because we don't basically, we are not really having the correct numbers at the moment, which are really maybe really the relevant numbers.
Absolutely understandable. Absolutely understandable, because it was for me also very difficult to find hard facts to introduce as a moderator to this actually at this forum. So I have still some concerns and some ideas that I want to discuss about, because being honest, I mean, 10 years ago, I've been to Shanghai. And that was not the time where we have had the pandemic, but most of the people would wear plastic masks, face masks to cover, to actually against the pollution. And after living in Graz, which all Austrians say that the Graz is somehow a cutter, and that's why the air quality is not good. On my opinion, after now living in LA, trust me, Graz is beautiful and has a great quality of air. In Austria, we would speak that the air quality in Graz is not good and we need to do something, blah, 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 blah. And then I went to Shanghai and I couldn't breathe. And I have no respiratory conditions and I couldn't really breathe. And I saw how many people are just in masks because of the quality of the air. So when we are seeing that, and we see also different kind of technologies coming up, and everybody is speaking about machine learning, everybody is speaking about artificial intelligence. So I want to challenge a little bit Zizi, asking what will be the technologies and what kind of technologies are nowadays already out there that can help us to investigate the problem and to maybe solve one or two.
Yes, so you what we've definitely seen in the last decades, and I don't think it's a trend that's going to reduce, is the increase of this hyper-polluted environment. And this is not just in China. No, the big cities, Shanghai, Beijing, of course, but they have the industrial belt area near Inner Mongolia where all the coal plants are, and that's also problematic. But you have New Delhi in India, and even closer to Europe, for example, you would talk about Lebanon because of the common electrical problems in the grid, because of the lack of infrastructure and war, etc., etc. You have a huge increase in electrical generators that burn all the time. So the air pollution at times is absolutely horrendous. Loads of examples around the world that have increased in time. And this is the outside environment that we can see and measure, and there are not, let's say, satisfactory, but at least standardized ways in which you can measure the air pollution and the particle size, and have some measure of comparison across. The problem that we find is that people don't spend all of their time outside. They spend a lot of their time inside their houses. And there isn't a sufficiently convincing method or standardized method to measure the level of pollution inside your house, because it's a different level that you would be exposed to. It's a different substance that you would be exposed to. So, for example, outside you would be probably exposed to I say outside and look outside, they would you would be exposed to a lot of pollution, like particles from exhaust, from burning fuel fossils, and from dust or from pollen. These tend to be a different type of particle than what you would find inside your house, which may be exposure to fungal spores if you live in a very damp place, or it may be exposure to material from clothing if the clothing is not appropriate quality or is fading. It's at the end of its working life. So the way that we can measure inside the house, I haven't been convinced we have a good standardized way. For measuring the pollution outside the house, we have a standardized way, but it's too crude. It doesn't let us differentiate sufficiently the types and level of pollution that we need to differentiate. And I can understand that a lot of the requests by policymakers, for example, where they would come and say, well, give us exactly guideline what we need to do, because they can see these numbers. They measure diligently, at least in Europe, every day. You have your pollution index, your pollen index. You can have a really nice dashboard. It's all very colorful. So what shall they advise? It's not sufficient level of detail. It has to be combined with other information to actually be actionable.
Absolutely amazing discussion. I'm really enjoying, although the whole topic is very scary, but I'm enjoying having this, you guys having the background and the information giving us. We have in question and answer part of the discussion, Professor Murthy, I hope I'm pronouncing the name right, who offers a collaboration, because he also wrote that he is working on that issue too, in microplastic issue, and he would be happy to collaborate, and spoke about gold nanoparticles based spectroscopy, which we have in Graz actually, and we do too. So I mean, yes, and thank you very much, Professor Murthy. And surface enhanced gamma spectroscopy is one of the technologies that can help us to actually spot the nanoplastics or maybe microplastics in the body, but it doesn't solve the problem that they are there, and we need somehow to deal in 3 different layers. One, reduce the exposure. Second, to spot them. And third, to either neutralize or take out of the body that they are not causing any harm. So coming from the Professor Murthy's comments, coming to you guys and Lucas, which organizations or which kind of researchers or which kind of policymakers or the society, who should collaborate in this? What are the key people that should collaborate together to make things better? Because we speak about the pollution since, I think, since the earth exists, and we think about the plastics. And I live in LA, and avoid plastic in my household is almost impossible, because I cannot buy anything without plastic.
Yeah.
So
Yeah,
how we should go? Who should collaborate? With whom we should collaborate to make it happen?
Well, I think the first thing is now that more people and scientists, you know, go into this kind of field to try to measure the particles, because that's important. And therefore, Professor Murthy, thank you very much. I think this is very important, because the detection of these particles in human tissue, for instance, or in any tissue, of let's say farming animals. If it comes to, to our food chain, it's called biomagnification, that we know that the higher the food chain, the more contamination you have to expect, and to define the contamination in this products, which come from, no matter if it's a vegetable or drinking water or meat or whatever, and to set guidelines. I think this is very important. So I mean, I'm trying to team up with people who are working, in food production and are scientists there, to try to define a threshold, which is basically something which the industry can work with, because I think this is essential to make the life easier for the industry, but also to help people being less scared, because I think we all are very, feeling insecure because we have no idea whether the use of this or this product is toxic or not, because we have no borderlines, no thresholds, no nothing at the moment. And I think first we need to define, and be able to measure these particles with high fidelity in different, tissues and different products, and then we can talk about, what and we have to, of course, going to medical research, therefore also the collaboration with different clinicians. And I'm in pathology, so we have a lot of, historical tissue samples where we have all the clinical data. We can correlate contaminations with clinical outcomes. So I think if we are able to measure particles, we can at least correlate a concentration of particles or a combination of different particle contaminations in tissues with clinical outcomes. That's what we normally do in pathology with, gene expression or protein expression, and then we can at least have an idea which kind of particles might be toxic, and then you can do more mechanistic experiments in cell culture or in animal experiments to recapitulate, what we find. And that was the original idea in this, our Micro-1 project to this. But we discovered that it's not so easy because, we don't have this kind of technology there. We collaborate with chemists and with analytical people who work with this kind of spectroscopic, like Professor Mercy in technology, which is essential to get the right numbers, because that's the big issue. And I think to bring these fields together, this analytical field, the medical field, and the chemists who we need to produce the particles which we can't buy, because the particles which come into our life by decay of bigger products, they are not produced by the industry. They are just around, and we need to so basically mimic these particles by producing them, and therefore we need the chemists to help us to get these particles in the right numbers, in the right sizes, and the right shapes, and so on, so we can work with to do our experiments.
If I may just add to this, I mean, absolutely in agreement. This whole idea of exposome that we often use at IR, it's a term that it's a blanket term. It's a collective name for all the exposures during a person's lifetime, and we simply don't have the tools that we require to do the study sufficiently well. Or well, for some aspects, perhaps. For most aspects, not a complete set. We've just coming to the end of the first ever round of big funding by the European Union, and second round of big funding by the US, in terms of developing tools for exposome research. And there is the International Human Exposome Network that has been funded by the European Commission with the participation of Japan, the US, and South Korea and some other partners. But that's where we are. We're still talking to one another. We're still discussing the tools, trying to provide the definitions and stabilize the definitions, at least in part. so that we can progress further. I absolutely agree with what Lucas has said.
Thank you very much for commenting on that. Coming back to understandable research, because I mean, research in politics and in policy making is one thing, and another thing is research or science that is understandable for many people. And actually, yesterday in HealthCom in news, it was an article, your tea bag might be shedding billions of microplastics. And a great picture of a nice tea bag with how much and how, and I looked through how they, I mean, they may be right, but they may be wrong too. And not each tea bag is like that as well. I don't like the tea bag because I like my tea really good one. Nevertheless, there are so many people who use that tea bag. And if you are in a restaurant, usually you will be served with a tea bag. And if we are speaking about even millions of microplastics in one tea bag, I don't know how they calculate it. I don't know how they measure to say in one tea bag millions of microplastics. But nevertheless, I actually, a human being, what we should do ourselves to not be scared and to try to avoid, independently of what is happening in surrounding. Before we come back to, actually we have 13 minutes left, and I would like each of you to give a wrap up or a direction. What do you think what the next 5 years or 10 years will bring in this research and policy making and change of behavior, and what we should do now to not come after 5 or 10 years and say we were wrong what we have done? So may I start with you, Lucas?
Yeah, thank you very much. So yeah, I think it's and already said this, so I think this is a field, at least what the human exposure or the exposure of mammalians is concerned. It's a field in the infancy, and we just discovered 2 years ago, you know, by colleagues from the Netherlands, that we have an average of what they measured, 1.6 microgram per milliliter plastic particles in the blood. So we basically transport it all the time into the different organs. So I think one of the things we need to do definitely is to find the risks for our health, you know, in different diseases, in different model systems, for humans to define the risk, you know. And that the good news might be, you know, that nevertheless we have this high concentration, you know, most of plastics in our blood. Most of us are still fine, but of course we see developments like early cancerogenesis, which you know, was just published 2 years ago by the by NCI, calling it you know, alarming, you know. And so we observe it as pathologists, and we talked about cancer, that some cancers, you know, like intestinal cancers or endocrine cancers, you know, come earlier and earlier in life, and we need to also diagnose them and prepare for having younger and younger cancer patients. So this is alarming, and we don't know why this is occurring. We have different stressors which could lead to that. So I think one of the big tasks also of the public is to raise awareness and to fund this kind of science, that we can, you know, elucidate the role of these particles and in the different you know, stages of life, and definitely also for the evolution, you know, because if we discover and we see that the particles can breach the blood-brain barrier, but also breach the placenta, and we don't know what happens in the development due to these particles, and the concentrations become higher and higher. There was just a publication I found yesterday in the which is not yet in bioarchives, that there's an increase in the measurement of the plastic particles in different organs after 2014 and 2024. So obviously, the increase in the production and then the increase in the decay leads to increase of measurable particles in different organs, which is actually quite logic. But it means that we have higher concentrations which we can measure. So we need to take care about that, and this is the one part, which is basically what we as pathologists always do, try to measure, different concentrations of, let's say, facultative pathogens in different tissues. And on the other side, we need to, take care about how we deal with plastic, especially when it comes to the food chain, how we can reduce the numbers of particles in the food we're taking in, like the drinks, the food chain, especially when it comes to meat or higher food chain products, which is definitely something which we should take care of first, and then try to set up thresholds of risky levels of plastics in let's say the water, bottled products, etc. So we can try to rationalize how we use plastic in our daily lives. I mean, and as Izzy said, indoors we have a higher pollution. For the plastic pollution, I can say it's about 5 times higher indoors than outdoors, and we have so many products indoors which we could think about to take care of, like our clothes, then the carpets, which many times have plastic fibers in them. So we can try to reduce those contaminations, because at least in Austria, people are indoors in average 23 hours of the day. They are indoors, and if I look around here in my room, in my office, I would say 80% of the surface is plastic. It starts with the color of the wall and ends up here with my chair and the surface of my tape and the phone and everything. So you can do a lot of things, by also feeling better by just reducing the plastic in your internal environment, which is, I think, also helping maybe your anxiety to reduce this kind of environment by reducing the plastic contamination in your personal environment. I think this is something you definitely can do. Yeah.
Thank you very much.
And I hope that is a little bit of an idea where in which direction it could go, but I apologize, it's very crude, but I think we need to think also in simple terms, because before we don't know how the contamination will continue, it's better to start with the simple things you can do.
Yeah, I mean, consciousness is actually the kickstart of doing something, if you understand something. By the way, I spoke yesterday to my students, and I told the pathologists are the mathematicians of the medicine. So now you proved my point with saying the pathologists want always to measure. So yes, that's how it is. Before I go to Thesis to ask him about the same question to wrap up, I want to just read Professor Murthy wrote, we could develop microplastics eating and the digesting microbiome. Maybe that can help us. So it's a great idea. I'm a little bit afraid of that, because if they digest microplastics, I'm very afraid they can digest ourselves too. Nevertheless, I mean, microbiome is everywhere. I can remember one of our colleagues from Graz gave a TED talk, and she started with bacteria are everywhere. in air, in ourselves, and we eat them, and when we die, they eat us. So I mean, bacteria are everywhere, and maybe we can use them for against plastics too. This is what you think about what are
I mean, I think from my side, the main need that I see is to raise the awareness of what this plastic pollution is. People think that, okay, they've replaced the straw in the coffee with from plastic with some pasta straw, and that saved the world. And it's not, and that shows the general lack of awareness. We have to look at things for our everyday life and everyday activity, a number of activities. Doing one thing is a token gesture that doesn't get you very far. I think for also just having a very open discussion helps. We don't want to be in a situation where everybody's just scared because suddenly they hear the very high numbers. The human body has a degree of tolerance. Lucas is absolutely correct there. But just being very open about how long the scientific process takes, what we're finding, why is it important. These arguments need to be articulated again and again and again every time new science comes to light. And I think it's just being persistent and consistent with the message. Other than that, it will come, but not in this generation. So if we're looking in 5 years, I don't think a lot will change. If we're looking at 10 years, if we start now, perhaps something will change.
I agree.
Okay, it's somewhat optimistic. So, because I expected a little bit more optimistic outcome, but it's somewhat optimistic at least. Guys, this was an honor to speak with you both about this, and I hope that we could raise a little bit of awareness. Professor Murthy, we will contact you, and we are hoping to have you to at our next discussion if we have on this topic. And I hope that we will have a little bit more positive things to tell, as not to tell your one tea bag has more microplastics as the measured yearly dosage. Thank you very much for the research you are both doing. And
Thank you for
I'm giving back to the forum, and we are open. Just drop an email if you have some ideas, and we are open to do research together. Thank you very much.
Thank you very much.
Thank you.